So now we've talked about inhibition. Now, inhibition can happen at the active site, as we've already seen, but it can also happen elsewhere on the enzyme. And when we are modulating or changing the activity of an enzyme by something binding somewhere else, we call this allosteric, or the allosteric site. And so the allosteric site is where inhibitors or activators will bind, and it is a location that is not the active site. Oops, that is a G. Let's fix that. So to understand this better, we'll talk through one specific example of allosteric control, and we will see this example pop up later this semester. So to give us kind of a case study, we're going to look at glycogen phosphorylase, also abbreviated as simply phosphorylase. So this is an enzyme that takes the glycogen in our body and converts it to glucose, right? So imagine you haven't eaten in a while, right? Your body doesn't have any energy from the food that you ate, but that's okay because your liver stores energy, glucose, in the form of glycogen. And this enzyme catalyzes the breakdown of glycogen in your liver to glucose 1-phosphate. Glycogen is basically just a polymer of glucose. On the right is a reaction scheme, folks. So you can see that this enzyme breaks down glycogen with n numbers of residues. To it, it adds a phosphate to make the products glycogen, which is shorter by one residue of glucose, and glucose 1-phosphate. And I've shown you the structure of glucose 1-phosphate here. This enzyme is modulated by a wide variety of small molecules, some of which are allosteric. So let's start with talking about this enzyme when it is not phosphorylated, because this enzyme is also controlled through phosphorylation. So the enzyme is in the T state or inactive state, generally when it is not phosphorylated, but it can be turned on by the molecule AMP, adenosine monophosphate. AMP is a low energy signal, and we will learn more about this later in the class in terms of why we consider AMP low energy. If we have very little energy, that is a signal to our body, hey, you better liberate some glucose from glycogen so you can make more energy, more ATP, right? That turns this enzyme on into the R state or the active state. Now the molecules ATP or G6P, ATP is adenosine triphosphate, G6P is glucose 6-phosphate. These are high energy signals, right? If we have a lot of ATP, that means we have a lot of energy. If we have a lot of G6P, that means we have a lot of glucose. There's no reason to get more glucose from our glycogen stores. Now these molecules, we call them feedback inhibitors because they are kind of downstream products coming back to inhibit. This is different than product inhibition, of course. Product inhibition is where the direct product is inhibiting. ATP, G6P, high energy molecules, they should inhibit this enzyme because we don't need to make more energy, right? Now, this enzyme can also be phosphorylated. So this is a way potentially to modulate an enzyme's activity. And when this enzyme is phosphorylated or a phosphate is attached to it, it turns the molecule, the enzyme, on into the R state, right? Now, even if the enzyme is phosphorylated, glucose, small molecule glucose, can again do feedback inhibition. Glucose is not the product of this enzyme, but it is a signal that we have plenty of glucose around, no need to take more from our glycogen stores. Now, bringing it all together, folks, here is the full picture that I'm showing you. Don't really worry about the phosphorylase A and the phosphorylase B nomenclature. Just think R state, T state. And so now you can see kind of the wide variety of ways that this single enzyme is controlled. Activated, inhibited through phosphorylation, dephosphorylation, activated, inhibited through the presence of various molecules. And what I want us to do, and we'll practice this in class, is think about which of these small molecules, ATP, G6P, AMP, and glucose, might bind in the active site to do competitive inhibition, or might bind in the allosteric site to either inhibit or activate this enzyme. And I want you to think about what we've learned about how molecules can bind to the active site, to think about this. To wrap up, folks, I'm going to kind of compare what we just talked about, activation and inhibition in the allosteric site, to structural changes, because the structure of the enzyme impacts its function, right? And in particular, I'm going to be talking about allosteric activation. So on the screen, to orient us, these are three-dimensional images of the structure of phosphorylase. On the left-hand side is the structure of the T state, and on the right-hand side is the structure of the R state. I want to point out the allosteric site of the enzyme, which is up here, and the active site, of the enzyme, which is down there. What happens when the enzyme is either phosphorylated or when AMP binds is we have a conformational or shape change where the N-terminus of the enzyme reorients itself to the top here of this picture. Now we know from hemoglobin that when one area of the enzyme structure changes, the entire structure will have some slight modifications because it's all connected to the primary, secondary, tertiary structure. Now, if this amino acid here, which is a serine, is phosphorylated, that's what you're seeing here in the image. If serine is phosphorylated, suddenly it's gained a negative charge, right? It went from polar neutral to negatively charged. This is a big difference. And so that negative charge then is stabilized and forming ion pairs with two arginines located here in the allosteric site. So this shape change allows the phosphorylated serine to be stabilized by arginines in the allosteric site. And when that shape change is happening in the allosteric site, a shape change happens in the active site. And that shape change in the active site here allows an arginine to reorient, a different arginine in the active site, to reorient to better bind one of the substrates of this enzyme, phosphate, right? Imagine that before the arginine was facing away, and now it's facing towards that phosphate, ready to bind. And so this is how an allosteric modulator can impact the activity of an enzyme. And just to convince you that the shape is changing of this enzyme, right, there are shape changes happening elsewhere. For example, this little green Turner loop disappears in the R state, which just tells us that the shape changed. And also, for example, this blue helix that's being shown here in the structure is in a totally different location. When shape change happens at one place, it happens throughout. And that wraps up our discussion of inhibition today. We talked out kind of looking at all the different Lineweaver bird plots and using them to identify what type of inhibition is happening. We talked through the Km and Vmax situation of competitive, uncompetitive, and mixed inhibition, at the same time discussing kind of why the Km or Vmax changing or not changing makes sense. Finally, we discussed allosteric control and talked through one specific example of how that works. With that, I leave you with this cat meme. Thanks, everyone.